The overall goal of the following experiment is to examine developmental effects of knockdown or overexpression of genes in specific subsets of neuronal progenitor cells in the neocortex. This is achieved by in vivo electroporation of DNA into rat embryos to transfect neocortical progenitor cells. As a second step, the electroporated region is dissected in order to enrich for transfected cells. Next, these cells are dissociated and plated in chamber slides and cultured in order to examine the effects of both intrinsic genetic alteration and application of exogenous factors. Results are obtained that show effects on neurite outgrowth and branching based on quantitative measurements using AxioVision software.
The main advantage of this technique over others, such as lipid-based transfection of primary neuronal cultures, is that we can target specific subsets of neuronal progenitor cells. For example, only glutamatergic neurons will be transfected using this method. In addition, we can electroporate at earlier embryonic stages and target early-born deeper layer neurons, or we can electroporate at later stages of development and target later-born neurons that are destined for more superficial layers of the cortex.
This method can help answer key questions in the field of neurodevelopment. For instance, how do intrinsic and extrinsic factors interact to affect neurite outgrowth and branching?
Although this method can provide insight into neuronal process outgrowth, it can also be applied to other neurodevelopmental processes such as synaptogenesis, cell proliferation, and cell-fate determination.
Prepare an injection needle as described in the accompanying text. Avoiding bubbles, backfill the needle.